Hello everyone. Today our Bioinformatics Capstone group will be presenting our work on creating the genome assembly of Poeciliopsis monacha. My name is Mariella Hartfelt.
My name is Rebecca Higginbotham.
I'm Kaiku Kaholoaa.
And I'm Teresa Marquez.
Poeciliopsis monacha, or P. monacha, is a type of freshwater fish that is also known as a headwater livebearer that resides in northwest Mexico in rivers, streams, and creeks. The P. monacha population consists entirely of females that are hybridogenetic hemiclones. A hybridogenetic hemiclone is an organism where during their reproduction process, half of their genome is passed to the next generation, while the other half is discarded. In the case of P. monacha, females mate with males from another species called Poeciliopsis lucida, but P. monacha only passes down their maternal genome to their eggs, while the paternal genome is newly supplied by the males each generation. This passing of the maternal genome in P. monacha is what makes it a hemiclone. P. monacha expresses a form of hybridization, which in this case is seen as the P. monacha and P. lucida genome hybrid that is maintained because of hemiclonal reproduction.
In order to better study P. monacha, we assembled multiple de novo genome assemblies. As a reminder, genome assembly refers to aligning and merging fragments from a longer DNA sequence in order to reconstruct the original sequence. So here, this image on the right represents your typical genome assembly where this original genome is unknown, and then you're going to sequence followed by the assembly of your reads to get your best estimate of that original genome. To briefly explain the construction of genomes, first you're going to isolate and sequence your DNA, followed by number 2, a quality control step, where you clean your reads by doing things like removing erroneous sequences. Then number 3, your assembler program will assemble your reads into contigs, which are contiguous sequences. In the middle section of this image, this is done by overlapping in order to create your final genome assembly. And once you have that assembly, you need to do number 4, quality assessment, which basically determines its completeness, accuracy, etc. But essentially, can we use it for downstream analyses? This leads us to our main research questions, which are 1, can we create the first genome assembly for this organism? And 2, of the multiple genome assemblies created, which assembler and genome assembly should be used to study the genetic basis of P. monacha? To begin, the assemblers, which are the programs we use to create the genome assemblies, included Hassler, Spades, Mazurka, Platanis Ali, and a hybrid pipeline, which utilized many different assemblers. For simplicity, we denoted these as Platanis A1, A2, and H1 through H6. In order to identify the optimal assembly, we use some statistics to evaluate each genome. The first is N50 and NG50, which, to put simply, are values that represent the length of our contigs. It shows how continuous a genome is, or in other words, are there gaps, are things missing, etc. A large N50 value means more continuity, which is desired because there are less gaps and thus less chances of error. Another statistic we use to evaluate continuity, not shown here, is the number of contigs in each genome, where you desire a smaller amount of contigs so that the genome is more continuous with less gaps. Next, we have KMR completeness, which helps us evaluate the accuracy of our bases. To put simply, we want to see if our KMRs have a high level of completeness and is representative of a haploid genome. In other words, we want our genome to represent a consensus haploid genome and not a diploid genome, which will also be further discussed in a later slide. Finally, we have BUSCO, which allows us to see how complete our genome assembly is. This relates to sequences conserved throughout all genomes. For example, organisms need necessary genes or sequences to survive, so those are going to be highly conserved across many genomes. This means we want a high BUSCO score, which means those necessary genes are present in our own assembled genomes, and is therefore one of the most important statistics when evaluating genomes. Finally, we have the genome length, which was not shown in the table, but it allows us to compare genomes created to each other. For example, if 4 genomes are similar in size, but one isn't, then we know that there could be something wrong with said genome. This can also be seen with the previously mentioned BUSCO scores, where these 4 genomes are mostly complete, but genome 4 is missing a lot of necessary information.
Moving on to our experiences with the various assemblers we applied, my group attempted to use both Hassler and Spades, but quickly ran into roadblocks in both cases. Hassler is a hybrid assembler typically used for long genome assembly, but we were unable to obtain results from this due to an age incompatibility with our software. We next attempted to use Spades, which like Hassler is a hybrid assembler, but is typically used for short genome assembly. We were unable to obtain results from this assembler as well due to a shortage of RAM.
Masurka is a hybrid assembler that utilizes a special type of read called mega reads. Unfortunately, these mega reads are large in size and thus storage inefficient. This assembler also has been running for 3 weeks and has still not completed its assembly.
Platanis LE is a de novo haplotype assembler that uses a phasing tool, which assembles each haplotype sequence in a diploid genome from beginning to end, and then pairs them as homologous chromosomes. Platanis LE is especially useful for analyzing highly divergent or heterozygous regions.
My group ran a combination of assemblers, including Platanis, Watchtower, DBG2 LLC, and QuickMerge, to generate multiple genome assemblies. In an attempt to improve the contiguity and completeness of our genome assemblies, we trimmed the data before using the assemblers. Then, using these assemblers, our group assembled 6 different hybrid assemblies and 2 non-hybrid assemblies. The 2 non-hybrid assemblies were generated using Platanis, which we refer to as Platanis A1 and A2. The difference between Platanis A1 and A2 is that Platanis A1's data was trimmed beforehand, while Platanis A2 used untrimmed raw data.
So here's a table of results from our Platanis assemblers, and in order to better assess it, let us first order by BUSCO score, as it was one of our strongest indicators of quality. And once we do that, we can go ahead and highlight some key pieces of information from each column. And to do so, I went ahead and color-coded the top 3 values of each column and added some reminders on the left side of the slide. And to summarize, we just want to see higher N50, NG50, and BUSCO scores while seeing a low number of contiguous sequences. And additionally, we also hope to see a total length that is somewhere close to the estimated genome size of P. monacha, which is about 590 megabases. So taking a quick look at this table, we can easily see that Platanis Elite dominated 3 of the 5 metrics for which we're assessing these assemblers. Taking a look at their N15, NG50, we can see that it was the least fragmented of all the assemblers and had the most functional completeness in terms of genes, given its 98.6% BUSCO score. And although the number of contiguous sequences isn't as low as we would hope, and the total length isn't as close to the estimated genome size of P. monacha as per se Platanis A2, it is still a strong candidate for a high quality genome assembly, especially indicated by its high BUSCO score. And especially talking about BUSCO scores, we can see here indicated by the blue that a majority of our assemblers reached a nearly 80% completeness, with an exception of Platanis H3, which only reached about 0.7%, and Platanis Elite that reached almost 99%. Given this and the results we just discussed in prior slides, we will only be focusing on our best assembler moving forward, which was a Platanis Elite, and we'll next assess its base accuracy.
This is a KMR plot, and when we're looking for visual representation for successful genome assembly, we look for plots such as this to reinforce the statistics we've gone over so far. So this plot measures base accuracy and completeness. It's comprised of 2 peaks. On the right, we've got a list of homozygosity, and on the left, we show the heterozygosity of the assemblies. So when we're looking at this graph, what we're hoping to see is high level of consensus. And in more traditional and model species, when looking at graphs like this, it's common to see higher peaks on the right and lower peaks on the left. That means we usually tend to see a higher level of homozygosity and a lower level of heterozygosity. But let's just review P. monaca for a minute. We're talking about a diploid organism. It contains two sets of genetic information, one from mom and one from dad. And when we create these assemblies, what we're looking for is high levels of agreement in the construction of the assembly itself. In this way, we're looking to create haploid data from a diploid species. The reason we tend to look for higher homozygous peaks is that high heterozygosity can lead to all types of noise and create chaos that can cause an assembly itself to fall apart. But here we have an excellently rated assembly that appears to just have high levels of variance. So perhaps we're seeing some pattern in the data here. P. monaca is a species that uses a hemiclonal method of reproduction. So that male genetic information is from a different species. And all that male information, based on previous studies, indicate that the male genome data is only used for reproduction and then later tossed aside. So perhaps what we're seeing is some level of integration happening between these two species, and we're just previously unaware of it. It could be that we're seeing some genomic crossover from the other species itself, or it could just be that using Poeciliopsis as an assembler that uses phasing to address these divergent regions was just better suited overall for this assembly. But despite the presence of heterozygosity, we were able to be successful in creating the genome assembly of P. monaca.
In this table, we can compare our P. monaca results located on the far right to 3 other de novo genome assemblies of fish in the Poeciliopsis genus, P. retropina, P. turberinus, and P. occidentalis. We can see that our results resemble previous results from other genome assembly papers, especially our assembly size, GC content, and BUSCO score, which gives us a degree of confidence in our assembly. It also suggests that the assembly can be used as a reference for other hybridogenetic genome assemblies.
Another takeaway we can take from the previous slide is that by having multiple genomes in the Poeciliopsis genus, we can use comparative genomic data to better understand some of the evolutionary processes that are inclusive to this species. And this is why large-scale genome projects, such as that of the Vertebrate Genome Project, or the VGP, are so important to the future of conservation biology research. The standardization of methods for genome assembly proposed by VGP are the same standards we have used throughout this assembly process and have been indicated several times throughout this presentation. The work done by these large-scale projects in creating open access genomic libraries that have high sets of standard for completion allow us to have high-quality data that can be used for future projects.
In conclusion, Poeciliopsis was the most efficient assembler, and we will be submitting a de novo assembly of P. monaca using this method. Thank you for listening to our Bioinformatics Capstone presentation.